So, welcome to this session on histology of the lymphoid system. Now, before we go to the details of the histology of the lymphoid system, let's talk some basics like what basically we are going to talk about the histology of the lymphatic tissue. Okay, and when we try to classify the lymphatic tissue is uh, classified as MALT, the mucosa associated lymphatic tissue. Okay, mucosa associated lymphatic tissue is seen in the GI tract, it is seen in the respiratory tract. Okay, and it is quite diffuse. Some places it is dense, and those places are Peyer's patches in the ileum. Okay, the lymphoid organs are divided into two. One is the primary lymphoid organ, where the lymphocyte synthesis takes place. For B lymphocytes, it is the bone marrow, and for T lymphocytes, it is the thymus. Okay, and the secondary is where these matured B and T lymphocytes go and they settle down. That are called as the secondary lymphoid organs, and they are the spleen, the lymph nodes, okay, the tonsils. They are the secondary lymphoid organs. Okay, and now we'll be talking about the primary lymphoid organ first, and the first organ which we are going to discuss is thymus. And thymus is considered its in production, proliferation, and maturation of T lymphocytes. And another interesting thing about this thymus is 90% of the T lymphocytes produced here. 90% of the T lymphocytes produced here are killed or destroyed by apoptosis because they are directed towards our own body, and only 5%, 5 to 10%, 10 to 5% are released in the bloodstream, and then they go and occupy various positions. Okay. So some things about thymus, how it is different from spleen, tonsil, that it is a primary lymphoid organ. Okay. It has, of course, it is the site where T lymphocytes mature, are produced, and they proliferate, mainly concerned with T lymphocytes. There are no reticular fibers. Okay, there are reticular cells. There are reticular cells, no reticular fibers. There are reticular cells which form what is called as blood thymic barrier. Okay. Now, how does a thymus slide look to us under a microscope? Okay. So under a microscope, this is suppose a slide. Okay. Now in this slide, the thymus is divided into various lobules. Okay, the thymus is divided into various lobules like this. So various lobules are there inside the thymus, and each of these lobules, now this is one lobule, this is one lobule, has an outer part which is cortex. So this part is cortex. Okay, so this part is cortex in this lobule. Outer part is cortex. Okay, in this part, outer part is cortex. So this is the cortex. And this is medulla, medulla of this lobule, medulla of this lobule, medulla of this lobule. Now what happens is the medulla become continuous with each other, and you can see here that medulla of this lobule is continuous with medulla of this lobule, it is continuous with medulla of this lobule. Okay, so here also there will be one more lobule like this, and this is the medulla. Medulla becomes continuous. And what is there inside the medulla? Inside the medulla, we see these reticular cells have degenerated now, and they are in the medulla like this, forming bodies, and these bodies are called as Hassel's corpuscles. Hassel's corpuscles. Okay, so in a slide of thymus, we now let us note down the key points. There are septae, which go from the capsule, which divide it into lobules. Okay, each lobule has an outer cortex, inner medulla. Medulla of various lobules is continuous with each other. Cortex has T lymphocytes which are developing, and there is blood thymic barrier. And medulla has Hassel's corpuscles. Okay. Now what are these reticular cells doing? So suppose this is a blood vessel over here. I'm now drawing a blood vessel. So suppose this is a blood vessel and the blood is flowing inside. And inside this blood vessel, there is the epithelial cells of the blood vessels. Okay. And this is the outer margin of the blood vessels. So the endothelial cells are there here. And the epithelial cells, okay, the epithelial cells which form the blood-brain barrier, they are like this. They literally clasp onto the blood vessel. So this is the blood vessel, and it's endothelial cells, and this is the wall. And outside these cells, they literally clasp the blood vessels so that, and here are the developing, these are the developing T lymphocytes. So it doesn't come in contact with blood directly. The nutrition goes like this. The nutrition passes through this blood thymic barrier to the T lymphocytes. This blood thymic barrier is very much necessary because these are developing T lymphocytes. Otherwise, they'll get sensitized against our own body parts. Okay. So this is the blood thymic barrier which exists very strongly in the cortex. Once the T lymphocytes are matured, they are checked only if they are those which are directed towards our own body are destroyed, and that's how 90% of those cells almost are destroyed. Then we move on to our next organ, and that is spleen. The spleen which in the fetal life was a site of hemopoiesis where blood cells were formed, and adult also it is site of hemopoiesis. So mainly it is concerned with destruction of RBCs which are more than 120-day old and abnormal RBCs. Okay, and of course is an important site for lymphopoiesis in adults. So in fetus, it is a site for hemopoiesis, that is formation of blood. And in adults, it is a site for lymphopoiesis. Okay, so fetus hemopoiesis and adults, it is site for lymphopoiesis. So how does the spleen slide look like? Now suppose this is a round, and the here also there is a capsule, And then there are trabeculae running like this. Okay, there are trabeculae dividing the spleen into various parts. Okay, and here also there are lymphatic follicles. In thymus there were no lymphatic follicles, but in the spleen what we see is around lymphatic follicles. Now these lymphoid follicles are there in lymph node also. So lymph node also you will be seeing follicles. So how do I differentiate follicle of us, or whether this is a slide of a spleen or whether this is slide of a lymph node? In many of these lymphoid follicles, you will see a arteriole. You will see a arteriole, which is eccentric. So that is the key feature of the lymphoid system. That there are lymphoid follicles, they are there in thymus also, they are in the tonsils also, they are in the Peyer's patches also. But presence of eccentric arteriole in the lymphoid follicle, is a key feature of spleen. So what we see is normally artery comes out like this. So I'll just draw artery comes out like this, and it gets surrounded by the lymphoid cells. Now I'm drawing an artery. Actually, the splenic artery divides and then arteriole comes out. And the moment it comes out, it gets divided. It gets surrounded by T lymphocytes first, and that is called as PALS, periarterial lymphatic sheath. Periarterial lymphatic sheath, which contains T lymphocytes. And then there is another layer called as PWP, peripheral white pulp, which contain B lymphocytes. Okay. So if you see this follicle, actually there will be two layers. One layer just adjacent to it, which is called as PALS, periarterial lymphatic sheath, which contain T lymphocytes, and peripheral white pulp, which contains B lymphocytes. Okay. Now what happens to these blood vessels ahead? These blood vessels, there are two theories that these blood vessels then open up. There is an open theory and closed theory. And suppose here is a venule, and then this venule ultimately forms the splenic vein. Okay, so artery is coming from here and vein is going out from here. Now what happens in between? There are two theories. One theory is that artery is directly connected to the vein, the blood remains always inside, and this is called as a closed circulation theory. Okay, and another theory is open circulation, where they say that the artery opens up and then the blood cells are free for some time and then they enter the venule by themselves. Okay, so this is called as open circulation. So there are these two theories in the splenic circulation. Now one more component about the spleen which we need to know, that we said how the blood passes through the artery and it is surrounded, that we discussed. Now the veins of the spleen also are, there are elongated cells called as stave cells like this. Now I'm talking about beyond the artery, what happens? There are splenic sinusoids. Okay. There are splenic sinusoids. Beyond the artery, there are splenic sinusoids, which are made up of long cells called as stave cells. And then there are reticular fibers running like this. That means there is a gap here. And through this gap, RBC comes out. Because of pressure, it comes out. If this RBC is normal, it again enters back. Okay, if the RBC is normal, young, it enters back. It's like Ali Baba. Ali Baba entered the cave and then came out. This normal RBC is like Ali Baba. It entered spleen and again came out, lean substance. But if it is like Qasim, Alibaba's brother, and it is more than 120-day old, then this will be destroyed by macrophages. Macrophages, which are around these splenic sinusoids, these macrophages, monocytes, and they are called as monocytes. They are called as, all these cells together, they form what is called as Billroth pods. Billroth pods. Okay. So now the spleen, the key features of the spleen now we will discuss. The spleen shows white pulp. Now what is white pulp? Nothing but a arteriole which is surrounded by periarterial lymphatic sheet and peripheral white pulp. So this is called as white pulp. White pulp has three components. It has an eccentric arteriole, it has a periarterial lymphatic sheet which has T lymphocytes, it has peripheral white pulp which has B lymphocytes. That is the white pulp of the spleen. Further, the artery divides, and then there is something called as red pulp. What forms the red pulp? The sinusoids of the spleen surrounded by Billroth pods. Sinusoids of the spleen surrounded by Billroth pods, together they form the red pulp. So red pulp, white pulp. These are the two identifying features of the spleen. Now we move on to the next organ, and that is the lymph node. Okay, lymph node is bean shaped or kidney shaped like this. Okay, there is a capsule, then there is a subcapsular sinus, and then here is the hilum, and then there are trabeculae. Okay, and this part where the trabeculae is called as cortex, and in the cortex, and this part is medulla. So this is cortex, this is medulla. In the cortex there are lymphoid follicles. Okay, now the afferent lymph comes from here. The lymph enters from here into the subcapsular sinus, goes through these follicles, and the efferent, so afferent is here and efferent is here. Also from here is artery and a vein entering the lymph node. Okay, so afferent, the lymph enters from here, from the capsule, and leaves from the efferent lymphatic vessel. So there are these follicles. Now these follicles are of two types. Some of these follicles show a germinal center. They are called as secondary follicles. Some follicles don't show a germinal center. Some show a germinal center. Some don't show a germinal center. Those who don't show germinal center are called as primary follicle. So primary follicle is an uninfected follicle. Moment it gets infected, it develops a secondary follicle. Because moment it gets infected, the antigen is taken at the center and new B lymphocytes are produced, which produce plasma cells to produce antibodies against the antigen. And therefore the center appears pale, which is now called as the follicle called secondary follicle. So which have a pale center are called secondary follicle, which does not have a pale center are called as primary follicle. So basically in the cortex are B lymphocytes. In the spleen, periarterial lymphatic sheet had T and peripheral white pulp has B. In the medulla, there are B lymphocytes forming follicles, and the T lymphocytes are in the paracortical zone. So where are the T lymphocytes? They are in the paracortical zone. While in case of the spleen, the T lymphocytes were in the periarterial lymphatic sheet just around the efferent article. B lymphocytes were in the peripheral white pulp. So from the primary lymphoid organ, they move to the peripheral white pulp of the spleen and cortex of the lymph nodes where they form the follicles. And T lymphocytes move to periarterial lymphatic sheet of the spleen and paracortical area, surrounding the medulla, the paracortical zone are T lymphocytes. Okay, so that is the lymph node. Now here, along this margin of the germinal center of a secondary follicle, are cells called as M cells or memory cells. They keep the information of the previous infection. Now coming to the last organ of this, and that is the tonsil. Mostly it is palatine tonsil. Now, how is tonsil different? Tonsil will also show follicles, but this tonsil, palatine tonsil, is mostly present. It is mostly present uh, if the not mostly, it is present in the oral cavity, and therefore it will show a epithelium. It will show a non-keratinized. It will show a non-keratinized stratified epithelium. Non-keratinized stratified epithelium. And it will show this tonsillar crypts. Otherwise, everything is like a lymph node. It will also show follicles. It will be showing follicles, some primary, some secondary follicles. Primary follicles are without germinal center, secondary follicles are with germinal center. Otherwise, the palatine tonsil exactly looks like a tonsil. Exactly looks like a lymph node. So basically, now how do we differentiate the various slides? How do we differentiate the various slides of the lymphatic tissue? Where follicles will be seen in spleen. No follicles, then now these are these slides, lymphatic organs, and of course there may be a gut associated also, where Peyer's patches may be there like this. So no follicles. If there are no follicles seen in the slide, we say the slide is of the thymus. Now, if there are follicles and there is an arteriole inside, eccentric arteriole, then that slide is of the spleen. Okay. If there are follicles and there is non-keratinized epithelium, then it is a tonsil. Non-keratinized epithelium and crypts. If there are follicles and then there is a gut, you can see the gut, the GI tract, then it is Peyer's patches. And none of this, and you see a bean-shaped follicles in a bean-shaped organ, then it is a lymph. Okay, so this, in case of infection, these organs, they enlarge in size, and that what is called causes lymphadenitis. Okay, the tonsils enlarge, the lymph nodes enlarge. So that's about the histology of the lymphatic system. We started with saying that there can be a mucosa associated lymphoid tissue, malt, which is diffuse, then there can be dense in form of Peyer's patches. Lymphoid organs, we said primary production of B lymphocyte. They primary produce lymphocytes. B lymphocytes are produced at bone marrow. It is a primary lymphoid organ. T lymphocytes are produced in thymus, which is secondary lymphoid organ. While secondary are from here, once produced here, they go and settle down in lymph nodes, spleen, tonsils, Peyer's patches. So these are secondary. Thymus, we said 90% T lymphocytes are killed and only 5% remain. It's a primary lymphoid organ. It is mainly concerned with production, proliferation, and maturation of T lymphocytes. No reticular fibers, there are reticular cells which form the blood-thymic barrier. They cover all the blood vessels. Why? Because we don't want them to be sensitized. There are septae, there are lobules, then there is cortex and medulla. No follicles. Okay, no lymphoid follicles. Okay, there are lobules, then there is cortex, T lymphocyte, medulla, Hassel's corpuscles. Okay, in the medulla we see Hassel's corpuscle. Medulla is continuous, and in the cortex, what we see is the developing T lymphocytes. In the spleen, what we see is lymphoid follicles with the eccentric arteriole. The arteriole is covered by lymphocytes. The covering immediately is periarterial lymphatic sheet containing T lymphocytes. peripheral white pulp containing B lymphocytes. Then the blood, there are two theories. One is open circulation and closed circulation, and the blood vessel further ends in a splenic sinusoid. The splenic sinusoid has long cells, stave cells, and reticular fibers. The RBCs can come out. If they are normal, they will again enter the sinusoid. If it is abnormal, it will be killed. Okay, so spleen, the characteristic feature is white pulp, red pulp. White pulp has periarterial lymphatic sheet containing T lymphocytes, peripheral white pulp containing B lymphocytes, while red pulp has sinusoids, which is surrounded by monocytes, macrophages, and reticular cells, and that is the red pulp. Then we discuss about lymph node, which is a bean-shaped organ. Afferent lymphatics come from the convex side. From the concave side is the efferent leaf. There is arteriole entering, venule entering. There are lymphoid follicles in the cortex. Medulla has macrophages and monocytes. And here there are two types of follicles: primary without a germinal center, with germinal center there is secondary follicle. Along this margin of the germinal center of M cells or memory cells, which keep the memory of the antigen. The tonsil also exactly looks the same, but there is non-keratinized stratified epithelium, and then there are follicles and there are tonsillar crypts. Even the Peyer's patches look same with follicles, but then we will see the villi. We see the villi of the small intestine. So lymphatic organs, how do we identify the slide? There are no follicles, it is thymus. There are follicles and there is the eccentric arteriole, it is spleen. There are follicles and crypts, then it is tonsil. There is follicles and GI tissue, it is Peyer's patches, and follicles and a bean-shaped organ, and that is the lymph node. Okay, thymus, the pathology, histopathology of the thymus, the problem which leads because of the dysfunction of thymus is myasthenia gravis, while lymph nodes, they enlarge in case of infections and even in case of cancer. That's about all about the histology or microanatomy of the lymphoid system.